is the organism you're looking at. Hello? Give me the answer.
It's beta hemolytic.
Beta hemolytic. Gram-positive coccus, catalase negative. If it's gram-positive coccus, catalase negative, it's immediately what? A strep or an enterococcus. So this is a strep or an enterococcus. Which one?
PY or G?
Pyogenes. And what group does that belong to?
It's group A strep.
Group A.
Okay. Good. But you can't say group A strep unless I ask, well, what group is this? If I ask for genus and species, you have to say Streptococcus
Streptococcus
pyogenes.
pyogenes.
Okay? Alright, here we go. Maybe I've had too much coffee. Alright, so now, what's the zone? No,
So it is
what
alpha.
color is the zone?
Clear.
It's green. So this is sensitive or resistant to the P disc. It's sensitive to the P disc. So if a gram-positive coccus is catalase negative, it is alpha hemolytic, and it's sensitive to the P disc. What genus and species am I looking at? Yvonne? She was like thinking about cockles. What genus and species? Britt?
Is that the staph?
It's a strep. It's a gram-positive coccus, catalase negative. Streptococcus pneumoniae. What group does it belong to? No group. Because it's got a capsule. And remember, it's the sugars that are stored in a carbohydrate. As carbohydrates, the sugars that are stored in the cell wall. What's the name of the scientist who figured out a way to group the streptococci based on carbohydrates in their cell walls? It was a lady.
It was.
We have a Rebecca in the house here. No, not Reinhardt. It starts with an L. Lansfield. That's correct. I'm sure you know her. She's important. Okay? Here we go. So is this particular organism sensitive or resistant to the P disc? Resistant. All right, now we have a purple color. So this purple color turned purple after 15 seconds. Is this a positive or negative? What test is this? Oxidase. Is it positive or negative?
Positive.
It's positive. And so if it's positive, is it an enteric or non-enteric? Non-enteric. If it's positive, is it a facultatively anaerobic organism or an aerobic organism? Aerobic organism. Very good. So the organism that's being portrayed by the positive, by the way, what is the reagent for the oxidase test?
Oxidase reagent.
The oxidase reagent, very good. So the organism that's being portrayed here as a positive is one that produces a bluish-green pigment on any media, and it also smells like corn tortillas. What is the organism? No, not Staph aureus. It's a gram-negative bacillus. It is a non-enteric because it's positive for oxidase. Pseudomonas what? Aeruginosa. Look at you. Daniel Simon.
Pseudomonas what?
Pseudomonas
Originosis.
aeruginosa.
Tortilla.
What?
Corn tortilla.
Because y'all did it as your organism? All right, here we go. Look, this is mannitol salt agar. Remember, if you know the name of the plate or the media, you know a lot about what's going on. Mannitol salt agar. What's the principal substrate of mannitol salt agar?
Mannitol, which is sugar.
Why is salt part of the name?
Because it has salt in it.
Because what?
It has salt in it?
Because it has salt in it. What percent salt is in it?
6.5.
No, not 6.5, take your point. 7.5. And why is it there?
To inhibit.
It inhibits what, Kimmy? Which bacteria? No, all bacteria except it doesn't inhibit halophiles. So it's going to inhibit every single organism unless they're a staph or a micrococcus, because those are halophiles. Yes? So if it grows on mannitol agar, it is a halophile. It's either staph or micrococcus. Nothing else will grow on it. Okay? Remember that. Okay, so it's selective, why? Because of salt, 7.5%. Be sure you don't just say salt, say 7.5, right? And it's differential, why? It's differential, why? It's turning yellow, why? That's another way of saying it.
Well, it's SRS.
pH indicator is what?
Phenol
red.
red.
But what's being fermented?
Mannitol.
Mannitol. Very good. Very good. So which organism is being represented in section 1 because it's fermenting mannitol? Staph aureus. It's the only one that we're working with that can ferment mannitol. Okay? Here we go. PEA, some questions will be quite easy. What does PEA stand for? Phenylethanol agar or phenylethyl agar, both of them are good. And what's the purpose of using this type of medium? It inhibits what? Gram neg, so the gram pos grow here, and it gives you an opportunity to acquire gram positive, especially if you have very, very aggressive gram neg. And there are some aggressive gram negs. Okay? There's also one very aggressive gram pos.
Dun, dun, dun.
All right, here we go, looky here, this is EMB. So in EMB, what's the principal substrate? Lactose. Remember, lactose is king for EMB, Hacto-1, and McConkey. If I ask what the substrate is for those three tests, it's lactose. Okay? On EMB, what is the pH indicator?
Also phenol red.
Eosin Y, or Eosin, I'm fine with whoever said Y is good, but Eosin is perfect. I don't need Eosin Y. If you put me Eosin Y, I'm gonna be happy, but Eosin is all I need. So why was methylene blue and bile salts added to the medium?
It's to inhibit.
Why was methylene blue and bile salts added to the medium?
Because it inhibits.
It inhibits certain
gram-
bacteria.
It inhibits gram positive. So the only thing that will grow on this plate and Hacto-1 and McConkey are gram negative organisms. Okay? What was the application for this medium? What is it used to test?
Fecal something in water.
That fecal contaminants in water. And if you just said water, I'd be really happy.
Poop in water.
What's that?
Poop in water.
Yeah. Poop in water. Okay. Yeah. And so if you see a metallic green organism growing on EMB, what is it?
Beautiful.
It is beautiful. Any questions about EMB? All right, this is McConkey. McConkey, which one, A or B, is a lactose fermenter? B, that's correct.
The big one.
So in EMB, crystal violet and bile salts are added. Why? Inhibit gram pos. What's the principal substrate in McConkey? Lactose.
Lactose.
And what is the pH indicator in McConkey? Neutral red. That's correct.
Neutral, that's the next different one.
This, did I miss one?
Yes.
No,
No,
not
you
yet.
didn't.
Okay. So wait a minute, let me check, cause I missed, oh no, here it is, okay, fine. I wanna be sure I represent it as much as I could. All right, this is a catalase test. What's the substrate in the catalase test? Hydrogen peroxide. What's the reagent in the catalase test? Hydrogen peroxide, very good. So, A or B, which one represents Staphylococcus micrococcus? A, right, so if it bubbles, that means that it has the enzyme that can break down hydrogen peroxide, and that's gonna be a staph or a micrococcus. Remember, in order to do the catalase test, you have to have a gram-positive coccus, right? It's the only thing that's important for it. If you put a gram-negative in here, it's gonna be positive every single time, and you're gonna be chasing phantoms. They don't exist, okay? So, which A or B represents Streptococcus or Enterococcus? B, very good. And true or false, you should always do a catalase test from blood agar? True or false? False. All blood has its own catalase activity, it'll give you a false positive. What type of tool, the substrate of the tool that you should use to actually do, a wooden stick is perfect, plastic, but never metal.
Non-conductive.
Metal will react with hydrogen peroxide and you get a false positive every single time. Okay? Very good. Okay, so this is the Staffy Tech. What type of test is it? It's not coagulase. Coagulase is another test. So we're looking for the coagulation of plasma. There's no plasma here. Yes, ma'am? It's an antigen antibody test. That is correct, right? So that's the mechanism of action, right? And which genus and species is Staffy Tech very specific for?
Oh, so it's like
Only
the
Staphylococcus.
blood one, the blood clotting
Doesn't,
mechanism.
it should react with anything else. Although in our test, there was some clumping with things that weren't supposed to clump. So the salesperson came to visit Myra, and what they found was the test was bad. So he gave Myra a new set and a little gift, so she was happy. But this is very specific only for Staph aureus. I will not ask you about the coagulase test because we don't use it here, right? This is so much better than coag. If you take this course at one of the other campuses, they use coag. Okay? We moved away from it. All right, this is the Kirby-Bauer test. Kirby-Bauer were the two individuals who came up with this test. The other name of this test is called the antimicrobial sensitivity testing. All right, so let's go through some of the terms you should know. What is this growth called?
It's a
A
lawn.
lawn. What is this clearing called?
Inhibition.
The zone of inhibition. Very good. What is the standard of turbidity that we use to make sure that we have the correct population to challenge the antimicrobial agent? Speak it, say it. McFarland, very good, very good. So the other thing is, so for us, I'm not gonna ask you to measure, we're just going to say, if there's a nice big zone around it, then we're gonna say it's sensitive without measuring it. It's not the best way to do it, but I'm trying to get concepts out of you, not perfect, right? So if it's got a big old zone, then it's sensitive. If it's got no zone, then it's resistant, yes?
Mhm.
Well, what's happening here? You have colonies inside a
Breakthrough.
cell. It's a breakthrough. So true or false, you should use CIX to treat infections.
No.
No.
False, not no, it's either true or false. You can't say no. Okay? Give two reasons why there might be colonies grow into the zone.
Is it-
Breakthrough is one, and the other one is what?
Could be it's partially
It's
resistant.
adaptive.
It's only resistant to a certain point.
That's the same thing as a breakthrough. That's correct, that's the same thing as breakthrough. Yep, anybody else? Huh? Could be overgrowth, but not typically. It would be, you have a contamination, you have two organisms, and the one that's growing inside of it is the one that's resistant to it. But the morphologies would be very good. Okay? Alright. Alright, so this is Hector and enteric agar, right? Did I miss one?
Yeah.
Okay. This thing is jumpy. So this is Hector and enteric agar. What's the pH indicator in Hector and enteric agar? Bromothymol blue. What is the principal substrate? What? Lactose, very good. So there is a presence-absence indicator in here. What is the presence-absence indicator for hydrogen sulfide? Huh? Iron, very good. So here, if we look at this colony right here, or whatever, because I was saying, we have kind of a blue-green background, and there's a colony, but it's got a dark center in it, it's black. So what is the results? There are two parts of it. What are the results for this colony right here? Lactose non-fermenter, you can't say negative, lactose non-fermenter, and it's
Yeah,
H2S
H2S.
positive, that's correct. So what pathogen is this representing? It's an enteric pathogen. Very common. Not E. coli. E. coli is typically not an enteric pathogen, it's mostly a urinary tract pathogen.
Mirabilis.
Which one?
Mirabilis.
Let me see.
P. mirabilis.
P. mirabilis. No, not P. mirabilis, that's mostly urinary pathogen.
Okay.
But although it does produce this reaction, but there's another pathogen that does this, starts with an S and with an A. Salmonella. Salmonella does this. Okay? Remember this, if you have an unknown that looks like this, there might be salmonella. Yeah? Alright, here is Hector and enteric agar also. What's the reaction here of these colonies? Lactose fermenter. That's all I'm looking for. You can't say lactose pos, lactose neg. I prefer you say lactose fermenter or not lactose fermenter. Okay? All right, be sure you remember how to look at bile-esculin 6.5, right? So with bile-esculin, how do you inoculate? It's a slant. SSSB. What's the principal substrate in bile-esculin? Esculin. What does a positive look like? It's dark, and the negative looks like it doesn't change color, it looks kind of amber-like. Let's look at 6.5 salt tolerance, right? It's a broth, it has 6.5% sodium chloride in it. When you inoculate it, what does a positive test indicate, or what does a positive test look like? Turbidity, and the negative test looks like?
Clear.
It's clear. Good. It's that easy. Okay, so if you have a bile-esculin positive, 6.5% sodium chloride positive tolerance, right? And it's a gram-positive coccus, it's catalase negative. It's, some people will say it's alpha or beta on blood agar. They can't make up their mind. But the two principal important things here are bile-esculin positive, 6.5 positive. What's the genus of the species of the organism? Or let's just say, what's the genus? Because there are two of them and you can't really differentiate them. What's the genus of the organism that's a gram-positive coccus, catalase negative, 6.5 positive, and bile-esculin positive? What is the genus? Crickets. Enterococcus.
Oh, it's at the top, right?
Enterococcus. What group does Enterococcus belong to? A, B, C, D, E, F, G, all the way to R. Which one? Kimmy? Vivian? Jasmine? About A, any complaint? So it's not A. Susanna? Huh? K, not K, but good guess.
I thought it was K too. Oh, D.
Which one? D? Correct. D is correct.
It was D.
What did you say? Alright,
There's
so
only two that were in D, right?
So, this is a little bit advanced. It's beta hemolytic. It's group D. Does anybody know the genus and the species? Streptococcus agalacti.
That's gonna be on the test though, right?
Huh?
That type of question.
Agalacti. So here are a couple ones to consider. A throat culture gave the following results: gram-positive cocci chains, beta hemolytic, catalase negative, bacitracin susceptible. What's the genus and the species? Streptococcus. What group? A. So anytime you see that A and you have a zone of inhibition around it, it's group A.
This is strep pio?
Strep pyogenes. Not pio, pyogenes.
Pyogenes.
Pyogenes means the former of pus. That's what it's called. Okay, here we go. A skin swab gave the following: gram-positive coccus, catalase positive. It's a halophile, ferments maltol. Based on that information, what's the likely genus and species? Step words. Name two types of media that you would use on your unknown and tell me why you use them. PEA, Kimmy? And so you're likely to pull your gram pos. And then another one would be what? McConkey. And why McConkey? It inhibits gram positive, gram negative growing. And what other beautiful piece of information do you get from McConkey? Lactose fermented or non-lactose fermented. So when you go in there today, as you get ready to, I'm going to show you all how to differentiate the organisms very quickly so that everybody will probably know what your gram negative is today as long as you get in there. It's very easy to do. Okay? Good? Cool.
47.
Yeah, so I'm not gonna ask that. One way want to consider using an NA plate. No, I'm not gonna ask that. Okay, here we go. So if it's negative, what is gram-positive coccus? If it's negative, what genus, what two genera? Or if it's positive, what two genera? Staphylococcus mycococcus. Very good. So if it's positive, it's a gram-negative bacillus. If it's positive for oxidase, is it enteric or non-enteric? Non-enteric. Is it aerobic or facultatively anaerobic? Aerobic. Good.
She got her stuff down, bro.
What's that?
Kimmy got it on lockdown.
Kenny got everything on lockdown over there. You wanna take the test now? Okay, so don't forget decarboxylase. Let's just review them real quickly. So the decarboxylase test, if we think about it, what three amino acids could be the substrate? Lysine, arginine, or ornithine. Very good. What's the pH indicator? Bromocreosol purple. There is mineral oil at the top of the meniscus. Why?
It helps make it.
To do what?
Make it
Make it
anaerobic.
anaerobic. If you have a decarboxylase test that does not have mineral oil on top, is it a valid test?
No.
That's good. What does a positive look like? Purple or violet. The negative looks like what?
Yellow.
Any other color. And then with the deaminase test, so this is a slant, how do you inoculate it? SSSD, very good. And so the principal substrate in the phenylalanine deaminase test is what? What? Phenylalanine, very good.
Phenol what?
Phenylalanine. This is how you spell it.
Phenylalanine.
Phenylalanine. It's an amino acid. It's a really important one. So when you come back in 24 hours, you add a reagent. What reagent do you add to the phenylalanine deaminase test? Ferric chloride. And what color is positive? Green. And what color is negative? Some not green, but rusty orange looking color. Okay? Okay. So here we have a gram-negative bacillus that's a lactose fermenter. It's oxidase negative. It has bile precipitate. What two organisms does this particular grouping of characteristics represent? What two genera? Huh? Not Enterococcus, thank you. This is the gram-negative bacillus. E. coli and Citrobacter frundi. Very good. Any questions? Don't go anywhere. I'm going to go get the UV stuff. I'll be right back.
Whoa. I feel super underprepared.
Yeah.
As far as knowing all that. The reagents, all that shit, I got it, but I gotta do what I always do. Crayon, memorize it over the weekend.
I totally forgot about this like exam, so I'm supposed to be going on a camp this weekend. I'm gonna bring my laptop in
Oh, you better study.
because I still have it. Huh?
Make sure
Yeah,
you
I'm taking
download
multiple
everything in
days.
case there's no Wi-Fi out there.
Yeah.
Download everything, make sure it's ready.
I'm
and
not
then-
going anywhere.
I'm gonna be studying.
If I don't have Wi-Fi, then I'm driving to the nearest Starbucks and
The
just.
kids are having their first.
All right, y'all come on over here. real
No,
quick.
it's not. It's something else. Some
So
of my friends.
this is my assay for the UV. This is Serratia marcescens on this side. That's Bacillus subtilis. They were actually, I did a Kirby Bauer. I made a lawn for everything. So I did it at 0, which is the control. I did it for 1 minute, 3 minutes exposure, 5 minutes exposure. Then I did 2 sets of plates at 10 minutes. One of the plates did not have the lid. The other plate had the lid. Okay? So can you appreciate that the controls and the plates that were done at 10 minutes with the lid look exactly the same? That's because the control showed what they look like when they're in their perfect state. They're not exposed. Here, they were exposed by 10 minutes, but they had the lid, and plastic or glass stops UV completely. So you can see how beautiful that lawn is on there. They're just completely covering the plate. The other thing to consider is with Serratia marcescens, that even after 1 minute, but probably at 30 seconds, I didn't do it at 30 seconds, you can see that almost everything was destroyed.
Yeah.
Yeah, except for, in some cases, at the very edges, you can appreciate that there is, even at 5 minutes, that there is growth. Not at 10 minutes, just because I didn't get lucky enough to get growth at that. But what hypothesis could you provide for the fact that you have growth on the edges of the plate? Shadows. So there was a shadow on the plate. The way that the UV light was shining on it, the plates are kindly curved. When the light hit, it provided a shadow, and whatever was next to the actual plastic was protected and therefore did not get destroyed. But everything else that it came in contact with got destroyed. So UV is effective by this particular instance at 1 minute, but probably at 30 seconds. But it can't be a sterilization method. Why? Because anywhere there's a shadow, the organism will survive. And so it's hard to not have shadows in things you're trying to sterilize. Yes? But it's an effective sanitation method.
UV
Okay.
is also dangerous then.
It is very dangerous to us.
Well,
It can
That's
burn
what
our
the right-
eyes. Huh?
Cancer.
So I said it has to be the right type of like
This is
strength of
UVC.
UV.
This
Yeah,
is
some
UVC.
people. I had a conversation with somebody. They were like, "Oh yeah, in Japan they have these things in the bathroom where you can sanitize
Sanitize your
your
hand,
phone
yeah.
and your hands." I was like, "Well,
And
if
even
it's true,
even if it's UVC, it's not going to be there long enough. But if you sit under UV light for 5 minutes, you're going to be royally sunburned.
And I explained that to them. I was like, "Yeah, if it's truly,
Yeah.
no."
But now something very interesting happened, Ruth, with Bacillus subtilis. It grew everywhere. Even at 10 minutes with the lid taken off, there's a lot of the organism growing on here. Can you provide a hypothesis for why that might be?
What was the name of the bacteria again?
Bacillus subtilis. Now provide a hypothesis.
Darn. I liked it and it grew.
Yeah. Provide a hypothesis. Yes?
We gotta think it through.
The what? No, I think you're playing.
You gotta think it through. What does UV do? It creates
It's attacking the DNA, right? So
capsule?
therefore, something is protecting the DNA.
It has
As
a
a
capsule.
capsule.
Not a capsule.
envelope.
Envelope?
Not an envelope.
Capsule?
It does start with the knee and end with the knee.
Oh.
Endospore.
Endospore.
Oh, that's amazing and scary.
So can you see
Bacteria is smart.
that UV cannot be used as a sterilization method
Okay.
because it doesn't kill the endospore. And by definition,
We just need Lysol.
I'm sorry.
well, that's sanitizing also, right? But at 10 minutes, it's pretty good, right? But still, look at all the organisms. That's why when Clostridium or Clostridioides difficile gets in an environment, it's hard to get rid of.
Just burn the building down.
Well, I don't know if it goes that far. I just have metronidazole available. So this will be, just like we talked about it, will be on the practical. Be sure you understand it. Okay? And it's explained by me on Blackboard under the UV. I have a little video, I think, of it. I'm explaining it. I know. I know. But to me, that's better than having, well, maybe not for Ruth, than having a book that you have to pay $240 for, right?
Yeah.
Your aseptic technique is not too great, I think.
No, mine is perfect. It's the tool that I used, right?
I heard mine was perfect too.
It was, yours was good. Mine was trash.
So I'm gonna keep these things. If you want to take pictures of them, you're welcome to do so. But I mean, I couldn't have asked for a better assay. This is beautiful. And I was doing this while everybody was asking me questions and all kinds of craziness.
Oh my God, somebody pat him on the back.